The sliding filament theory of muscle contraction is a theory that proposes that our muscles contract when some filaments slide over each other. And when I say it's a theory, I don't mean it's a theory like my theory about who will win Top Chef this season. I also have one of those. But this theory is more like the theory of general relativity that Einstein used to explain gravity, or evolutionary theory from Darwin. I mean, these are theories that have mountains of supporting evidence. We're pretty sure this is how muscles contract. But to understand this theory, we need to understand a couple other things. First, we need to understand the concept of excitation-contraction coupling. Contraction, we know what that is when the muscles shorten. Excitation refers to your nervous system creating an excited signal to tell the muscles to contract. But how does this nervous system signal, this action potential, zap a muscle and make it do stuff? That's tricky to do. So that's what we'll look at in this video. In the next video, we're going to look back at striations. We've been talking about those since Unit 1. Oh, they're just little lines on some types of muscle tissue. But what are those? Because if we can really get deep down on striations and understand their form and function, then combined with our understanding of excitation-contraction coupling, we'll have a good idea about how this sliding filament theory works. So let's get started by looking at excitation-contraction coupling. This is taking place at the neuromuscular junction. That's where a neuron meets a muscle at a junction. So this is where the excited signal is going to somehow jump into the muscle tissue, zap it, and cause it to contract. So let's start by looking at this junction. At the top of my image, I have a neuron reaching down towards, at the bottom of the image, a skeletal muscle fiber. Now there's a small space in between these called the synapse. So much of what we'll discuss is what's going on at this synapse. Now this all begins in skeletal muscle when you decide voluntarily to make the muscle move. So you initiate an action potential, an electrical signal, an excitation that will travel from your central nervous system down towards the muscle. So I've just indicated that as a lightning bolt here in our image. You know, the axon has ion channels that open. Ions are flooding into the axon, causing this action potential to occur. We don't need to get into the details of action potentials right now, but we want to understand that that signal will eventually reach the end of this axon at a structure called the axon terminal. Now, inside this axon terminal are vesicles. We talked about vesicles back in Unit 1. There's a lot of different types, but these vesicles are called synaptic vesicles. They're full of neurotransmitters, and what I want you to see here is that they will drift towards the bottom of the axon terminal, fuse with the cell membrane of the neuron, and dump their contents out into the synapse. When we say contents, in this case, when you have a neuron talking to a muscle fiber, the contents will be a neurotransmitter called acetylcholine. Acetylcholine's pretty cool. It's the first neurotransmitter ever discovered. Now, we're keeping it pretty basic here, right? But even just this is a pretty amazing feat. We've taken this electrical signal and we've converted it into a chemical signal, which is now drifting inside of our body across this tiny, tiny, tiny space. So what happens next? These neurotransmitters are floating out there, and they will eventually reach receptors after crossing the space called the synaptic cleft. So the neurotransmitters hit these receptors. They're not going to enter the muscle fiber. The neurotransmitters will just attach to the receptor for a period of time, and then they'll detach and drift away. But when they attach, and if they attach in sufficient numbers, what can happen is that this excited signal, this action potential that's now turned into this chemical signal, can trigger ion channels in the muscle fiber to open, and ions start flooding in just like they did in the neuron, and we trigger a new action potential, a new wave of electrical activity traveling down the cell membrane of this cell. So that's pretty good. We took an electrical signal, we turned it into a chemical signal, and we reinitiated that electrical signal on the other side of the synaptic cleft. But if we stop here, all we're doing is zapping the superficial portion of the cell. In other words, this signal right now is just traveling down the cell membrane. We need to get that signal deep into the skeletal muscle fiber. We do that by allowing that action potential to travel down these tubes called T-tubules. And to get an idea of what this looks like, if you took a muscle fiber, they're like long cylinders, and you imagine taking a pin and just poking little holes in the cell. The hole would be coming in perpendicular to the cell membrane, forming the shape of a T. So that's what these T-tubules are. They're holes and passageways perpendicular to the cell membrane, and the action potential can access the deep parts of the cell through the T-tubule. And as we follow our image down, we can see that the T-tubule reaches some type of structure here, which is called the terminal cisternae. A cistern is like, I don't know, a spittoon? Do you know spittoons? Like old-timey jars that people would spit into? Because spittoon. Well, a cistern is also like an old-timey word for some storage receptacle. And terminal means at the end, right? Terminator, John Connor, you terminated, And so we're at the end. We've reached this storage unit. And the storage unit is part of a bigger structure called the sarcoplasmic reticulum. This sounds very fancy. All it really is, it's the endoplasmic reticulum. But we're in a muscle fiber, and sarco means meat or flesh, which is what muscle is. So this just means the endoplasmic reticulum of the muscle cell. But what the sarcoplasmic reticulum does is store loads and loads of calcium. And as that action potential travels down the plasma membrane, deep into the T-tubule, hits that terminal cisternae, and the signal spreads into the sarcoplasmic reticulum, calcium is released. All throughout this muscle fiber, calcium is released. And that's really good for reasons we don't know yet, but it's important for calcium to flood into the cell. And once we understand what striations are, we'll see that there is a calcium binding site in and amongst the structures that make striations. And only when calcium can access that binding site can contractions happen. So we'll do that in the next video. But a final word here on this concept. Of crucial importance is this synaptic cleft. The events that take place within the synapse are very important. Not just in neurons meeting up with muscle cells. Like if you've heard of serotonin and dopamine and SSRIs, selective serotonin reuptake inhibitors like Prozac, those medicines and those neurotransmitters are all involved in regulating humans' mood. So lots of antidepressants are acting on these synapses and the behaviors of the neurotransmitters and the receptors and some enzymes and what happens in there. And so that's true also with acetylcholine. Acetylcholine imbalances seem to be playing a role in Parkinson's and Alzheimer's and other pathologies. Also, if you really understand this stuff, you can get into some trouble with stuff like creating VX nerve agents. And no better place to learn about those than the greatest movie of all time, The Rock, starring Sean Connery and Nicolas Cage, where they keep the world safe from VX nerve agent. But this is real stuff and serious stuff. If you recall, the dictator of North Korea, Kim Jong-un's brother, was killed when these two women ran up behind him and rubbed something on his face, and they said it was for a game show, and it ended up being VX nerve agent. And that stuff messes with what happens in the synaptic cleft. When that acetylcholine floods the synaptic cleft, it's supposed to bind with receptors, let the muscle contract, and then it detaches, and there's an enzyme that comes in and gets rid of the acetylcholine. What happens if you don't get rid of it? Well, that's what VX nerve agent does. It messes with that enzyme. You've got all this acetylcholine that's just now it's hanging out in the synaptic cleft. So your muscles are contracting and contracting and contracting again and again and again. So you're spasming until all that shuts down. All your muscles are fatigued, including your diaphragm, and then you asphyxiate due to what's going on at these synapses. So just a few little real world, except for the movie The Rock, real world applications of just this simple concept. Interesting.